It's the first three-dimensional human brain tissue grown in a laboratory, and it could help revolutionise the study of neural disorders like Alzheimer's and autism at their earliest developmental stages. The pink flashes are firing neurons, signs that viable brain tissue has been generated. Lead researcher Jürgen Knoblauch of Vienna's Institute of Molecular Biology says he's excited by his team's achievement.
The tissues that we have generated are not functional brains. What they are is they are developing brains. We have already used it to understand a rare human genetic neurological disorder that is called microcephaly. But this is a rare disorder, and for the future we hope that we or other labs will be able to use this system to model more common diseases.
The tissue evolved from human stem cells developed with nutrients and grown on a gel scaffold to form what are known as organoids. Up to 4 millimeters in length, the organoids, here swirling in a test tube, include parts of the cortex, hippocampus and retina. One region not present is the cerebellum, which is formed later in human embryos. The organoids could never form any level of consciousness and are far from fully functioning brains. But Knoblauch thinks they're perfectly suited to test the effects of drugs on humans.
Drugs and toxic chemicals are tested mostly in animal models, but then transferring this knowledge to humans, asking how would they actually act on human tissue, is incredibly difficult. And this is actually the biggest threshold in pharmaceutical research. And these organoids, whether it's our brains or whether it's the gut organoids from other labs or the eye organoids, will allow scientists to do experiments directly on human tissues.
Models of other human organs like eyes and livers have been grown in the lab before, but Knoblauch and his partners at Edinburgh University think this is even more significant. They hope these organoids will open doors to new areas of research and solve some of medical science's deepest mysteries.